My friend has
Oh wait, you
You don't have an
you don't have an HDMI, right?
Oh hey.
Oh hey, hey Max.
No problem.
No problem. I do have an HDMI. What do you mean? I do have a connector. I can.
Yeah, can we
Yeah,
connect?
can we connect?
Oh wait. I
Oh
think
wait.
this should be
I think
okay.
this should be okay. This. Yeah.
Because connect here and then it's a bit better. Or just move this. It should be okay.
Okay, yeah, I go there. Yeah. You're good?
Yeah, it should be good.
So the mic is not on yet, right?
Yeah, I have it here. I turned it off.
Okay, cool.
If you, I think if you have time now, it's better.
Okay, how are we doing? Time?
Yeah, it's fine. We can wait like 5 minutes.
Yeah, we'll have to connect, as I say, with one of my companies is basically focused on chaperones. It's quite interesting. We could exist now. We can raise. Is there something just on the left? I mean, human life. If it wasn't you want to have a special way of doing things, which is clearly best. We'll see if there can be possible. So you've got several companies running on it.
Well, we do. So basically, what happens is that
Yeah, I managed to. Cool, we have a few people on YouTube. So when we live stream to YouTube, they might have preference to watch it rather than come in person.
The last stream.
Yeah.
Okay,
But
I see.
you managed to reach a bigger audience. So.
Yeah, that's good.
we are still expecting a few people to come in. So if we can delay a bit.
Is it still out of term time or is
Term time just started.
just started?
It is.
So maybe some people haven't come.
Yeah. It is like exam season though.
No, but I think last time it was people come in a bit later. So
Yeah.
we can wait a bit.
So yeah, we can wait 5 minutes.
Yeah, I think 5 minutes, and we got a couple more people from my side that said they would come.
Tony, hello Tony.
So how long generally is a good idea to go? I think I'll probably be about 50 minutes. Is that
50, yeah, that's fine.
I try really not to go more because then it's people get start to fade.
Yeah, 50 is good, and then 10 minutes for Q&A.
Yeah.
Yeah.
Hi, how are you?
Pleased to meet you. Nice to see you.
Tony, and so Cambridge Clinical Labs and they're doing a lot of really interesting, innovative things. So this is actually the presence of the longevity.
Brandon,
You have to know Tony.
Yeah, do you used to be called Florence? She's at Trinity working on Parkinson's. She's at for second name. May. She's a postgrad. In fact, she's postdoc. She's postdoc.
Okay, no, don't know her.
Okay, she'd be interested. So pass that on.
Right. There will be recordings. So
You can, we have a live stream link. We can send her the live stream link if you like.
I shall speak to her afterwards.
yeah.
She's away. See you later.
Yves Baptiste.
Nancy's active as usual.
Did you fly a couple of days ago?
Yeah, I actually flew in yesterday. I just had it turned out to be very busy as usual before the talk. So,
Right.
So I came in. I was going to do a bunch of meetings, but then I came in and was like, my talk is not really ready because it's a bit different. This is a bit different talk than I usually do, and because there's a lot of investors and partners, or not like the full story, So I actually prepared a bit.
Right.
Thanks for organizing.
Yeah, no problem.
It's just it's really a lot of work to put this together.
Yeah.
Yeah, thanks for kind of.
Your team also helped.
Yeah.
Are they the ones coming to give the audience?
Yeah, I think there's some that might come in a few minutes. Yeah.
You actually met him in Darwin, kind of a garden party, not party, but like the college really is connected to you.
Yeah.
Cambridge is small and not so small, so it depends on. You can say hi. Yeah, I think we can start anytime. I think so, but we can wait 5. Yeah, let's sit until 5 too.
Yeah. Okay, I might start.
Just get this out of your way. And then
evening, everyone. It's a pleasure to welcome you all tonight, and an even greater pleasure to introduce Dr. Gergely Toth. Dr. Toth is not just the visionary founder of Cantabio Pharmaceuticals, which is a pharmaceutical company at the forefront of developing precision therapeutics and diagnostics for neurodegenerative disease, but also an expert with over 2 decades of dedicated research into Alzheimer's and Parkinson's disease. So Dr. Gergely Toth embarked his journey by earning his PhD at Creighton University in the US in 2001. His quest for knowledge then took him into the halls of UC Berkeley, where he refined his research as a postdoctoral fellow in the Department of Molecular Biology. Gergely then worked at the innovative biotech company Protein Mechanics, which was sold to Locus Pharmaceuticals, before leading a team at ELISA Pharmaceuticals working on Alzheimer's disease. From 2010 to 2015, Dr. Toth sharpened his expertise via a Wellcome Trust Research Fellowship at the University of Cambridge. So today he continues to share his valuable and pioneering insights to the academic and scientific communities as an honorary associate professor at UCL. So ladies and gentlemen, please give a warm round of applause to welcome Dr. Gergely Toth.
Thank you very much for this nice introduction, and thanks everyone for coming. I can see it's a hardcore Parkinson's disease therapeutic diagnostics interest audience I have here, so thanks. And also I want to thank obviously the Cambridge Majority and Aging Society for inviting me to talk about our research at Cantabio Pharmaceuticals, really targeting DJ1. With that, let me jump right in and talk a bit, really start very far away, just to give a bit of perspective of our vision with our therapeutic approaches targeting DJ1 and developing a disease-modifying therapeutic for Parkinson's disease. So we are very much interested in redox homeostasis, and I think most of you know what redox homeostasis is, but in fact, as we talk a lot about oxidative stress and its essentially negative role in the onset of diseases, but in fact, oxidative stress comes from essentially reactive oxygen species, which are fundamentally very much required messengers that actually control a lot of basic fundamental processes in cells. And these reactive oxygen species, when they are at low concentrations, they are imbalanced with kind of antioxidant enzyme systems in cells, and then we get redox cellular homeostasis and everything works fine. Now, as we age, or perhaps certain due to environmental factors, lifestyle factors, or disease, this balance essentially comes out of balance and results in cellular dysfunction, damage, and cell death. To look into this a bit more, we can look under the hood what's happening, right? So we got various reactive oxygen species. Think about hydrogen peroxide, superoxide, nitrile oxide, and so on. They're actually sourced from mitochondria and from various other enzymatic systems. They're controlled by, again, antioxidant systems. Now, at low exposure or at low concentrations, they actually control various redox signaling networks. And again, if everything is in order, then we got normal function. However, when this is disrupted, we have oxidative stress or oxidative distress, and various things go wrong. Now, again, it also has a number of positive functions. functions, right? So stem cell renewal, proliferation, healthy immune response, and even longevity. On the other side, if those levels are high, then we get stem cell exhaustion, tumorigenesis, autoimmunity, senescence. And in fact, what we are really interested in is neurodegeneration. And in fact, in Parkinson's, it is well known that oxidative stress is a very important, critical process that drives disease onset. So why is that? Well, there are certain genetic defects, there are certain mutations in certain genes that actually end up increasing oxidative stress through various mechanisms, environmental toxins, and of course aging. All can result in various biological processes that increase reactive oxidative species. Okay, so for example, one of the most important ones in Parkinson's is actually mitochondrial dysfunction. Mitochondrial dysfunction results in high oxidative stress. Another thing that's really specific to Parkinson's disease is actually dopamine metabolism and dopamine oxidation due to high oxidative stress. Now, high oxidative stress in neurons are particularly vulnerable to oxidative stress, and in different ways leads to neurodegeneration. I'll give you two examples. One is protein homeostasis. In Parkinson's, actually, significant oxidation drives significant elevation and Lewy body formation, or actually self-defense, essentially anti-stress defense, is also damaged by oxidative stress. Okay, so these are all specific to Parkinson's. I have a problem with accessing. Now, to dive into this a bit more, in redox signaling, there are certain proteins that are responsible for redox signaling through interacting oxidative species. These proteins, the function of these proteins are actually controlled by reversible oxidation. I'll talk more about that. Some of these proteins are oxidoreductases, transferases, hydrolases, lysases, isomerases, and ligases. So there's actually a fairly high number of proteins that are redox active proteins. The way this actually happens is quite complicated, and I don't have time to modify it and oxidize in various ways, very sensitively. Now, when oxidative stress increases, as you can see here in the red, these cysteines can actually oxidize, forming sulfonic acid, and some of this sulfonic acid is actually an irreversible part. Irreversible and essentially inactive form, where sulfonic acid can be essentially actually reversed. And so these modifications have a very particular function in redox signaling. Now, in oxidative stress and high oxidative stress, a lot of times what happens is these superreactive oxygens, superreactive cysteines, actually over-oxidize, get inactivated, and essentially these enzymes are not functional anymore. And so oxidative stress, a lot of times, and high oxidative stress, the main damage that it does to cells is not necessarily structural damage. It reacts randomly to different side chains, causing essentially protein structural destabilization, but rather very specifically knocking out these redox signaling enzymes. And essentially that's how the nature of the damage of oxidative stress happens. Now, this is kind of the background where it started from. And Cantabio pharmaceuticals, our vision was, okay, well, there are these essentially processes like oxidative stress that really cause damage to cellular function. But in fact, it's not just oxidative stress. There's other reactive stresses, for example, lyocell stress, and indirectly, for example, even protein aggregation that causes oxidative stress. That causes damage. And as we age, these actually have built up, and can cause onset and progression of a number of diseases, for example, Parkinson's, Alzheimer's disease. And so we are very interested in reducing these stresses. The question is, what essentially had been done was to reduce these stresses in a focused way. So, oxystress, you need antioxidants. Protein aggregation, you need aggregation inhibitors. Now, this is great, but the problem is that these stresses are very connected. So, oxystress increases protein aggregation causes oxystress, stress causes oxidation, oxystress, inflammation, which causes more oxidative stress. So, what we need is approaches where we actually have a systematic response to these type of stresses, which is actually not that easy. Now, it happens that we have a target, DJ1, which I'll talk a lot about, which actually can maybe be modulated in a way to counteract these stresses, because DJ1 is a controller of redox and protein homeostasis. And I'll explain what I mean by that. But before I do, let's look at kind of Parkinson's, Alzheimer's, and pathology. It turns out that let's say biochemical stress is very much a main pathology of these diseases. Think about Alzheimer's disease, obviously everybody has heard of amyloid beta, alpha-protein aggregation results in neurofibrillary tangles, alpha-synuclein aggregation results collapse in Lewy bodies, and so on and so on. And so we know that, and we can detect that, for example, with very specific imaging agents. And from these, and now with some breakthroughs in Alzheimer's disease in CSF, essentially IBDs, looking at essentially biomarkers of let's say tau phosphorylation or A-beta levels, we can actually see and diagnose these pathologies. And so what we learned from all of this is that these pathologies actually are present way before the symptoms arise, possibly 10, 15, 20 years before. So if you can see in the diagram, you got clinical disease stage. So from here you have symptoms. Essentially, both A-beta and tau aggregation and pathology, peripheral deposition, actually happens way before that. And neurodegeneration is happening. If you look at oxidative stress and glial cell stress, it's actually very similar. These oxidative stress and glial cells are very apparent very early on. The challenge is that we have a very good ways to measure actually both glial cell stress and oxidative stress in the brain at this point. So that's a challenge. But from postmortem brain examinations, we know that there's a lot of parasitization, the methylation, and products in both diseases. Let me switch to DJ1 and why we are working on DJ1. As I mentioned, DJ1 is a control of redox and protein homeostasis. And one of the reasons why I started working on DJ1 is because it's also genetically validated target, meaning means that mutations accumulated in DJ1 end up causing familial Parkinson's disease. Very simply, if someone has a very specific mutations that causes loss of structure, loss of function of DJ1, he or she will get Parkinson's, unfortunately. Okay, so that's a very strong validation that DJ1 is likely involved not just in familial Parkinson's, but actually in idiopathic Parkinson's disease. Now, DJ1 is a protein that is a homodimer. It's actually a small protein, and you can crystallize it, and you can recruit it from biophysical structural point of view. So it's amenable for small molecule discovery and all kinds of drug discoveries. So that was a plus factor for a small company like us to really try to tackle this. One of the caveats, however, is in terms of redox activated redox control protein. It has a very reactive system that's controlled by oxidative stress. And in fact, there are essentially no drugs out there that control and target proteins that are kind of redox, it's involved in redoxing, that can properly modulate this redox modulation of these proteins. So it's a very novel target from a therapeutic point of view. So what does DJ1 do? Well,
when it's synthesized or expressed, it has a biochemical function. It's also a very strong chaperone, so it has an enzymatic function, which I'll talk about. It's also a chaperone. It's a transcriptional factor. It's actually expressed in mitochondria. It enables mitochondrial function, and it's a transcription regulator. What happens is when there's high oxidative stress, you believe BG1 oxidizes, moves in the nucleus, and initiates an anti-stress response by overexpressing proteins that can counteract oxidative stress, and also chaperones that limit protein aggregation. So it does a lot of things, and in fact, it also involved in signal transduction, enabling self-reliable pathways to be activated. So a lot of times, when I talk to, for example, investors, they also say, well, what does BG1 do? And I say, well, it does all of these things. That's the advantage of it. It does everything just a bit. And that's all. And it's a very powerful way to possibly counteract a very complex disease. And so, that's an advantage and a disadvantage, obviously, from a neurodistory point of view. Now, because of that, it's relevant to a number of oxidative stress connected diseases like Parkinson's, diabetes, stroke, and even infertility. And these diseases have a lot of function. So from a therapeutic point of view, we want to increase its activity. While in cancer, tumor cells figured out that BG1 is a cell survival protein. How the cell survives, they actually decrease its level, and its activity. So in cancer, we want to knock it out and make it totally the opposite. Let me dive into a bit more about how BG1 is actually controlled by oxidative stress. So BG1 has a reactive cysteine, which is cysteine 106. It's in a pyruvate form. It can easily oxidize into sulfuric acid form, which I'm going to call this oxidized BG1 in this lecture. And then at prolonged high oxidative stress levels, it can over-oxidize into sulfonic acid form, which I will talk called over-oxidized BG1. We know that this is enzymatically active. We know this is enzymatically active. I'll show you some data on this. This likely has an effect in neuroprotection, high oxidative stress, and this is a pathological state because this is an unstable state, in fact. Now, we make BG1 in the lab, right, recombinantly, and we can actually oxidize or reduce BG1. Here, as I showed here, this is basically mass spec, or that's a proteomic study where we're looking at following oxidized and over-oxidized states. So we can oxidize by the hydrogen peroxide. We actually can fully oxidize BG1. And when we add high levels of hydrogen peroxide, the over-oxidized state will actually appear. So we studied in detail, from a drug discovery point of view, we really wanted to understand the structure. We really structure-based drug design, so we wanted to understand the structure. And that's fundamentally also very important from a signaling point of view and protein interaction point of view. And what's interesting is that if you look at the crystallized both the reduced and oxidized BG1, basically the same conditions, and what we found that overall the structure of reduced and oxidized BG1 is basically exactly the same. So that was interesting because, okay, if the structure is the same, how can they have different activities? And look at and essentially how can they localize in different compartments of the cell differently? But when we looked at it closely, we see that there are small differences in the active site. And so maybe that's the difference. Another thing that's really interesting is that oxidized BG1 terminally is much more stable. So the whole dimer itself is more stable compared to reduced BG1. We also see that over-oxidized BG1 we showed with biophysical methodologies that it actually is very prone to aggregation. It's stable. And we've done multi-dynamic simulations where we looked at essentially the free energy of the complex for reduced oxidized and over-oxidized. And what we can see is that actually over-oxidized is less stable. And most of this due to the fact that the third oxygen somehow destabilizes the thermodynamic interface. So let me kind of switch gears and talk about what happens to BJ1 in Parkinson's disease, because that's really important to understand in order for us to actually target it therapeutically. So what can we learn from essentially immune mutations? One thing that we know that most of the immune mutations actually, again, cause structural instability or actually protein never folds. So there's no active DJ1. So in this case, for example, a study on a patient with DJ1 L172 mutations showed that there's no staining for DJ1 in post-membrane tissue. So there's no active DJ1. The second thing that we know is that actually there's very strong Lewy body pathology when you with DJ1 mutant patients. Okay, so phenotype is aggregating, and so that's very, very important also, because also, let's say Parkinson's have semi-clinic pathology, but not necessarily, all patients. Another patient study showed this is a T154K mutation that is actually mitochondrial dysfunction and high oxidative stress. And no DJ1 can be measured in a blood test by ELISA. Okay, so in summary, what we know from these is that we have essentially high cyclic pathology and mitochondrial dysfunction, high oxidative stress. DJ1 kind of goes back to all the basic functions that I think I've been telling you. Now, in idiopathic patients, it's less unclear what's happening, right? Because it's much harder to essentially tell. But we know from one really well-done study is that actually in the brain, in post-brain tissue, is a significant loss of DJ1, total DJ1. The second study by Japanese group showed that in erythrocytes, in blood, there's actually more oxidized DJ1, cysteine, oxidized 6 oxidized DJ1. And that's actually very significant, although this is a small study. So this is what we know from human studies. Now, we've been working with DJ1 for 10 years, and we do a lot of biochemical studies. And as some of my colleagues are working with DJ1, they noticed that the SNAP protein tends to aggregate, and it's hard to work with sometimes. And so we started looking at this, and a study came out of this, which we published recently, in which we showed that actually DJ1 can misfold, polymerize, and actually form these fiber-like insoluble deposits. And when we looked at this in more detail, we saw these are fiber-like, and actually, based on this, we actually looked at what could be happening in post-membrane tissue, which I'll talk to you in a minute. So first, we just look at just some very basic things. We say, okay, well, are we losing soluble DJ1? And so we can't quantify soluble DJ1 as a function of time. And we saw that we were using some soluble reduced DJ1, but actually also we were using a lot more oxidized DJ1. So DJ1 seem to aggregate. Okay, so what kind of aggregates are we getting? Well, it turns out that these are tyrosine T positive aggregates, and most of you know that tyrosine T stands for beta sheet. Structures, for example, amyloid neurofibrillary tangles, also are identified with tyrosine T. And then we use FDR spectroscopy, and we showed that actually there's high, and then as neutron aggregates, there's a significant increase in beta sheet structure, both for reduced oxidized or oxidized. CD studies essentially supported this, where we could basically use ultracentrifugation, looked at the palette and the supernatant, and saw that the palettes are fully beta sheet-like. So all of this suggested that DJ1 actually, in fairly close physiological conditions, can form hemoglobin T positive fiber-like structures. And we said, wow, okay, we thought this was a very stable protein. How's this possible? But okay, maybe this is at fairly high concentrations, and this is an artifact. Well, let's look at the brain. And so, in collaboration with clinical neuroscience, we partnered with Roger Barker and his team, and Freddie Cantabio, we looked at this team. We looked at Parkinson's and Alzheimer's brain tissue slices. And here we stain for fibrils using PFDA, so you get the green fluorescence. Use an oxidized BG1 antibody. We've seen for nucleus. And so if you kind of look at this diverged genome, you can see that you got a stronger stain for oxidized BG1 that colocalizes with fibril. This PFD stain suggesting that where there's a lot of fibril structures, there's also oxidized BG1, and none of this happens essentially in control. Okay, well, we looked at phosphoserine, meaning Lewy bodies, right? And we saw interestingly that there's actually co-staining also, but it's essentially BG1 is kind of oxidized BG1 is around these phosphosynchronous is not really co-localizing, but likely in Lewy bodies around synuclein. And what was the most interesting that actually oxidized BG1 really co-localized with phospho-tau, essentially neurofibrillary angles. And we've seen similar things in Alzheimer's disease for tau, and in fact, we saw some polarization in beta amyloid. Perhaps this is not that surprising because actually in amyloid in Lewy bodies, there are other 400 proteins. We know that it's not clean fibril structures, but there's a lot of other proteins that go aggregate. And probably BG1 also go aggregate. So really the question now, is this a co-aggregation and what actually does BG1 actually aggregate by itself? So this is something that you're very interested in exploring, and we're doing some work on this. We don't have the answer yet. But one thing, just to kind of summarize, what's really important for us from these overall studies, we also can do a semi-quantitative analysis which suggests that actually there are increased oxidized BG levels in Parkinson's and Alzheimer's disease, and that's actually very important. Kind of going back to redox, homeostasis idea. Okay, so that's it. And if you look back at the literature, there's a lot of hints about BG1 aggregation, but no one has actually taken and explored it in as much detail as we have. So BG1 has been observed to present in high level aggregates in Parkinson, brain tissue. It's also in neurosoluble fractions in Parkinson's brain tissue. Actually, there's a Korean group that a long time ago published that actually BG1 aggregates, but their aggregates were not fibril necessarily, but actually they suggested that BG1 gives its structure, and then there's been a number of publications showing that essentially total GJ1 and synuclein co-localizes and co-localizes. So there's a lot of hints really supporting kind of our conclusion that actually BG1 aggregates and oxidizes, and this is very much part of the pathology of Parkinson's disease. Okay, so I mentioned that BG1 also has a chaperone function. And in fact, for over 10 years now, there's been several studies that show that BG1 can inhibit alpha-synuclein aggregation. And in fact, there's one or two studies that actually show that only oxidized BG1 inhibits aggregation. So that's, again, a very interesting phenomenon. Reduced BG1 doesn't seem to interact with BG1. So we were very interested in understanding this in more detail. And so we did a follow-up study. We showed that actually looked at the interaction between monomer synuclein and BG1, and we found that only oxidized BG1 interacts. And using NMR spectroscopy, we identified that actually the N-terminal of synuclein interacts with BG1. It's actually quite transient interaction in NMR, but when you do the biophysics, this is a protein interaction. So the KDPs, three different methodologies to show that the KDs under 100 nanomolar quite strong interaction of ribosome with the protein. And in fact, we have, and so, okay, this is a monomer DJ1 and oxidized DJ1. So monomer alpha-synuclein and oxidized DJ1 interact. But what happens in an experiment when both proteins are in aggregating conditions? And so you use dynamic light scattering experiments to probe that. With dynamic light scattering, we can really probe and measure early oligomer formation. And from all this what we see that oxidized DJ on alpha-synuclein can essentially inhibit oligomer formation. And oligomers only start occurring after 70 hours of the air in these conditions. In these conditions, when 70 hours, we starting to see oligomers. And if you use DJ1 and alpha-synuclein over oxidized DJ1, we actually see aggregates appear in the first few hours. So this is a massive delay of oligomer formation and inhibition of oligomer formation. So this is a very, very interesting system and suggests basically that when oxidative stress increases, DJ1 oxidizes. And in these disease conditions, when alpha-synuclein is more likely to aggregate, that's only when these proteins may actually meet in cell and neurons and result in essentially stabilization. Then we talk about alpha-synuclein aggregation. But if we go further, that actually is a very unique case because we're not just talking about synuclein aggregation here, but we're talking about DJ1 aggregation. And when you put this together, neither synuclein nor DJ1 actually aggregate. So this is beneficial for both protein systems. Let me switch gears again and talk a bit about DJ1 enzymatic function. So DJ1 has a number of enzymatic functions. So it's a big protease. It's actually quite strong esterase, and it's probably a fairly big glycosylase. But glycosylase function is actually also very important because essentially glycosylase is really part of the pathology of Parkinson's Alzheimer's. So what is glycosylate stress? This is not so much in the mainstream, right? So glioside stress is basically gliosides are reactive aldehydes, right? That can just interact randomly with protein side chains and cause, oxidative stress, inflammation, and hypoxia. They actually cause the mesglycylation end products. Mesglycylation end products generally causing inflammation and then oxidative stress. Now, methylglycoside is one of the most abundant glycosides in our body, and most of methylglycosides is actually a byproduct of glucose metabolism. So if you've got high glucose levels, you're going to have higher glycoside stress. Actually, glycoside stress is very important in diabetes too, and vascular complications of diabetes. Now, in Parkinson's, again, this hasn't been mainstream, but in the last few years, there's been really good work showing pathology of advanced glycation and products in Parkinson's brain. And also, have been shown that methylglycoside reacts to alpha-synuclein and causes its oligomerization, causes toxic oligomer formation. And DJ1 can degrade methylglycoside and other glycosides and actually form lactate. Lactate is generally protective at low concentrations. Now, we've been very interesting in life, so because we said, okay, well, DJ1 does a lot of things. Maybe this is the main function of DJ1. And so we've been looking at this from various angles. One way we made us, we are a pharmaceutical company, so we developed some inhibitors, some protein-specific inhibitors, and actually we developed covalent inhibitors. One of our novel covalent inhibitors inhibits glycoside activity at 900 nanomolar, which is not super active, but it's enough as a tool compound to look at a couple of things. And so one of the things that we've done with this inhibitor is we tested it in a glycoside toxicity assay. And in here, what we're doing is you're adding glycoside to cells. These are neoplastic cells. We have wild type in red, we have knockout in green, and then we have overexpressed vitamin B4 is actually DJ1 overexpressed in cell lines. What we can see is there's a huge difference between the cell viability and essentially the cell's resistance to glycoside stress. DJ1 overexpressing cells can really essentially are less sensitive to glycoside stress. And then wild type and knockout is just a little bit more sensitive compared to normal cells. And then we can take our inhibitor, and what's really interesting is these are essentially we basically use one concentration of glycine, is actually 4 millimolar here. And when we treat with our inhibitor, we basically knock out DJ1. You can see that the cells, depending on the doses of inhibitor, we see essentially cellularity goes down. So as we knock out DJ1 with our covalent inhibitor, the cell zone essential ability or defensive against glycoside stress really goes down, and therefore ends up essentially in cell death. Now, obviously, this type of therapy wouldn't be good for Parkinson's because it would essentially cause Parkinson's. However, this could be very risky if it's very specific against cancer, because we want to knock out DJ1 because DJ1 is involved in essentially cancer onset and proliferation in many different ways. I'm not going to go into that today. I want to focus on Parkinson's and neurodegeneration. So another very relevant, so another thing that we've really pioneered and looked at is, okay, well, what happens if we oxidize DJ1? Well, in fact, when we oxidize DJ1, we lose enzymatic activity, which is not a surprise because we modify the helic active side of DJ1 and still very important to doing sensitive function. But this is really interesting because I've been telling you that actually this is a normal function of DJ1 to get oxidized at low levels of hydrogen peroxide. Maybe it takes up new functions, but it loses its enzymatic function. And in fact, essentially oxidation of cis-106 completely changes the function of DJ1, we believe. And also, as I mentioned, location and localization. Okay, so just focus on Parkinson's. Actually, there's a number of other metabolites and things that interact with DJ1, cysteine 106. For example, in Parkinson's, Parkinson's honestly is the degeneration of the substantia nigra, the dopamine system, where dopamine is generated, right? So what happens is when you generate dopamine can react very easily with hydrogen peroxide and also stress to cause these very rapid dopamine prenodes. And in fact, probably that's why oxidative stress is very specifically relevant to the dopamine system, because if you increase oxidative stress, you got dopamine oxidation causes toxicity, mitochondrial dysfunction causes more oxidative stress. So it's a vicious cycle. Anyhow, so interestingly, oxidized dopamine can react with cysteine 106 and inhibit DJ1. And it's not just inhibits DJ1. If you look at the thermal stability of dopamine reactive DJ1, actually there's a loss of structural stability and thermal stability, and actually dopamine, oxidized dopamine modified DJ1 is very prone to aggregation, but it does not cause fibrillar aggregates. It just causes amorphic aggregates. You still lose your structure, you still lose your DJ1 activity. So again, this is actually a very specific, so oxidized dopamine modified DJ1 is very specific. Parkinson's relevant product, and it's probably be a great biomarker if you could measure it easily. So this is something that we are very interested in also actively looking at. Just to kind of finish my introduction, this is there's a number of studies that have been shown in vivo and mice in US vigilance and the target for Parkinson's. I'm not going to go into that, but I guess one important thing is that you knock it out in mice and also in rats and get Parkinson's like neurological symptoms. And if you overexpress it, then it's protective enough for Parkinson's models. Now, at Cantabio Pharmaceuticals, we're focused on DJ1 very much. And we have small molecule programs, we have enzyme replacement programs, we have a platform where we can measure DJ1 and oxidate DJ1 very precisely in blood and tissue lysates. And so I'll talk a bit about some of the applications here and therapeutic approaches that you're doing. I also mentioned that we also a very interesting protein is folding and have our own approach there, which I will talk about today. But actually, a lot of DJ1 work on stress and protein aggregation is very much linked to Toth and A-beta. And so these projects are at the end, and these works, this research is very much interactive. Okay, so one of the things we're doing, as I mentioned, if high oxidative stress, each one is more likely to use structure. And so one of the things that we've been working on is developing these what we call pharmacological chaperones. Very simply, compounds that bind to DJ1 and enhance structure. And in fact, this translates to longer lifetime in the cell, longer activity. It's actually a very complicated mechanism. Nevertheless, what we did here was to say, well, let's look at this the simplest way. Let's get reduced DJ1, oxidized DJ1, look for binders of small molecules. And we did this in a screen called chemical microarray screening. And idea is that we got a chemical microarray, small molecules are mobilized. You got DJ1 added to it, and when it's a binding event, you can detect that it's also a problem of resonance. So we're going to do pharmaceuticals with this, and screen 110,000 compounds, got a bunch of hits, very different scaffolds. I'm not going to go into this, and just to say that we pursued a number of small molecules here, and we developed one compound, one scaffold in more. This is called CD125 that interacts with DJ1 potently, about 200 to 300 nanomolar. It stabilizes the protein. It's an allosteric side binder. It doesn't want to because inhibit DJ1 and so it functions. And then, interestingly, it has various and fairly high activity in Parkinson's and Toth models. So, for example, in a cell model, the EC50 is about 220 nanomolar. That's something in a Parkinson cell model is all mitochondrial dysfunction, high oxidative stress, Parkinson's models. And actually, interestingly, it's also active in a proteasome inhibition cell toxicity model. So it is, we went further, and when we were developing, we actually developed a brain penetrant orally viable compound, and this was active in an MPTP mice model. I probably just cruise through this just to say that, in fact, this compound is very active, meaning that when we treated MPTP, mice with MPTP, this loss of essentially dopamine neurons, the same way with THR plus neurons, and this compound can completely eliminate that. And also, essentially, with some dopamine loss and full dopamine loss. So this is very promising. I'm really honest, we still have a long way to go to understand its full mechanism. But this is something that obviously works, and we're really pushing it forward. Another therapeutic approach is more straightforward, but equally challenging due to other things. But as I said, in Parkinson's, we want to increase DJ1 activity. How can we do that? Well, we can just inject more DJ1 or somehow, or express DJ1 in vivo. Now, honestly, it's not so simple because DJ1 is a site intracellular protein. So when we inject it, it doesn't cross the membrane. So what we have done, and this is not our idea that people are doing this, we create a construct where we added a cell-penetrant peptide to its N-terminal. So we get a homodimer here, creates a cell-penetrant peptide. And in fact, luckily, this actually takes it and gets into the cell, and actually cell penetration is quite efficient. Now, this doesn't work with every protein. It works with small proteins, as and other peptides. With larger, more complex proteins, this approach doesn't work. So, just to say is that what's really just nice about our drug candidate is that it has exactly the same structure as the ATP-G1. We crystallized both, overlaid it, it's exactly the same. And in fact, we've done some hemodynamic simulations some time ago. And why this is interesting is because essentially the idea is, okay, does the cell-penetrating peptide part, the yellow part, does that interfere with the active site? Does this interfere with any protein interaction site? We think it doesn't. This is a hemodynamic simulation. You can see that essentially the self-antigen peptide is actually very flexible. Here, this is the active site here, and it does not interfere with any of the main binding sites, we believe. So that's a good sign. Now we looked at its cell penetration in different cell models. This is the DJ1 knockout cell model. These are HEC-293 cells, and we added administered cells DJ1 as cell penetrating DJ1 using for histochemistry. And in DJ1 you can see some background. This is just sticking to the plate. While here you can actually see cell penetrating DJ1 actually getting into cells. And we used our redox DJ1 platform to really characterize its concentration and kinetics. So when we add cell penetrating DJ1, very quickly we see high levels of cell penetrating DJ1, and after 24 hours that actually reduces. And that makes sense because cell penetrating DJ1 will get degraded. Half-life of DJ1 is about 24 hours in the cell. When we do DJ1, we have no lesion in the cell, so it does not get no lesion gets to the cell. Cell penetrating DJ1 gets to the cell. So we have also looked at this in several Parkinson's models. So one is a paraquat model. This is again micro dysfunction model. So cell penetrating DJ1 is protective here. And actually one of the most interesting models and simplest models is actually the lifestyle stress model there. We add negative lifestyle to cells. We completely heal the cells. But if we completely do cell penetrating DJ1, we can essentially rescue cell biotic loss. Simply we degrade negative lifestyle directly and essentially reduce toxic lifestyle levels. However, again, oxidative stress and glycosylated is really not necessary in mainstream Parkinson's research. It's all about significant aggregations, you know. So everybody wants to see alleviation of significant aggregation of drug candidates, so these can modify therapeutic. In fact, as I mentioned, DJ1 is a good chaperone inhibitor of alpha-synuclein aggregation. And in fact, this cell-penetrating DJ1 is an awesome inhibitor of alpha-synuclein aggregation. So we have alpha-synuclein at 100 micromolar, right? In this assay, we got the blue is showing alpha-synuclein T, fluorescence indicating that synuclein is aggregating, and then we add cell-penetrating DJ1, we completely inhibit aggregation, and we can inhibit already at 10 micromolar. So substoichiometrically, 10%, if you go down, substoichiometrically 10% of DJ1 will inhibit alpha-synuclein aggregation. And it makes sense because it stops manipulation. It stops manipulation of, and so very low levels can have a drastic effect at initiating, just postponing nucleation events. And so we're very excited about this and really kind of focusing on essentially protein inhibition strategy with cell-penetrating for Parkinson's. Okay, so I'm getting to the last thing that I want to talk about at this point is the fact that, okay, well, we're building these therapeutic projects, but in fact, it's very important to measure the DJ1 levels and oxidize DJ1 levels because it helps to understand, we need to understand mechanism. The other needs to understand there are some other drugs, candidates are targeting DJ1 in vivo. And so we started building various methodologies to quantify DJ1 because these are not, you can buy a total DJ ELISA kit, but you cannot buy an oxidized or oxidized or reduced DJ ELISA kit. So we developed conformation specific oxidized specific antibodies and used for essentially reduced DJ1, oxidized DJ1, and over-oxidized DJ1. Let me correct myself. We couldn't get antibodies for reduced DJ1, so we designed our covalent inhibitor to be a local probe, and we built these sandwich ELISAs with this. And the idea here is that in healthy cells, in healthy people, you have mostly reduced DJ1, maybe a bit of oxidized DJ1. But as disease comes along, you have high oxidative stress, DJ1 gets oxidized. So you get less reduced DJ1 and more oxidized DJ1. pretty simple. And perhaps this oxidation will actually increase as disease increases. And so we have this technology working, so we call this redox regional analyzer platform. And so we have a couple of validations here. So these are cell models. So these are two cell models. One is very actually parasite and neuroblastoma cells, or we have 6-hydroxydopamine. Both are basically very simple Parkinson's models, cell models. And so we look at total DG1 dysfunction system. Green is control, red is 6-hydroxydopamine treated cell. You can see that there's in this model it's a loss of total DG1, which is quite interesting. That recapitulates, what we see, what people, you see other researchers talk, and sporadic Parkinson's, meaning there's loss of total DG1 levels in both brain tissue. When we add hydrogen peroxide, we actually don't really change total DG1 levels. And then these are DG1K cow levels, so essentially, maybe not DG1 out. So this is basically kind of the background here. Okay, so let's see what happens to oxidized DG1, reduced DG1. Well, in oxidized DG1 levels will go up a bit in the 6-hydroxydopamine treatment. That also makes sense because essentially we got oxidative stress generated by mitochondrial dysfunction as 6-hydroxydopamine don't go any and it's causing high oxidative stress, and essentially hydrogen peroxide addition, of course, will increase oxidized DG1, and exactly the opposite happens in reduced DG1. So you've got loss of reduced DG1 levels, 6-hydroxydopamine, and loss of reduced DG1 hydrogen peroxide levels. So this is great because fundamentally what we want to see, we kind of see this in cell models. So what we're doing now is we're looking at post-mortem brain tissue. So we've looked at healthy brain tissue, and what we see is that there are total DG1 levels, and essentially there's a little bit of oxidized DG1 and there's a lot of reduced DG1. We also looked at, now we really focused on blood tests, so we're looking at plasma with this, and so we can see that total DG1 levels in healthy volunteers, are vary quite a bit. Oxidized DG1 levels are a bit more constant, and also reduced DG1 levels vary quite a bit. But and then what we can see if you look at the total levels that reduced and oxidized and reduced kind of equals total levels, which also makes sense. And what we're trying to do with this is why this is exciting, because we like to think of this as a potential precision diagnostics. Why is that? So we are looking at the ratio between oxidized and reduced. And we can also control this by total DG1. So there's a lot of difference between age, younger people will have less DG1 or possibly more DG1. We don't know, we have to look at this. Older people may have more oxidized DG1. So their total DG1 levels could change depending on age and sex, right? So being able to control it is an advantage. And so again, in the future, what we're doing, we're looking at Parkinson's portion brain tissue and essentially Parkinson's patient's plasma in big studies to essentially do proof of concept studies that in fact this platform could be used as a diagnostic for Parkinson's disease. So just to say that, in fact, if you didn't know, Parkinson's is way behind Alzheimer's disease. So there's no blood test, there's no imaging probe. It's very difficult to diagnose Parkinson's disease. It only happens when people have severe symptoms, and at that point, 60% of dopamine neurons, 80% of dopamine neurons are gone, degenerated. It's very, very late. So it's very hard to also develop a therapeutic approach. So there's a lot of work obviously happening here, and the main type of biomarkers that people are looking at is mitochondrial dysfunction, also stress, obviously protein aggregation, phosphosynuclein, aggregation. Obviously, DG1 is very much related to also stress, mitochondrial dysfunction. Now the question is this really specific to Parkinson's? Because as I mentioned, this is the DG1 is involved in diabetes, infertility, cancer, infections, even sepsis. So maybe it's a lot more complicated, and it is a lot more complicated. But we are looking at, obviously, we got programs going on in diabetes, Parkinson's, very much also in male infertility. We'll be looking also into cancer soon. But what we are imagining from a business point of view is also important for us in a company that we may actually use this as a precedent for a general screen. Because with on the market, this is also very useful. You're 50 years old like me. I would want to do a redox ELISA screen to see how my stress levels are. And maybe you get a fingerprint from our aspects, and you can say, well, from depending on how your ratios come up, that could be indicative of certain diseases. I'm not going to go into this. So this is what we're trying to do with this, other than really use this as a precision help guide our essentially patient selection and precision therapeutic approach. So hopefully with all these, I have given you enough talk about believing that we could actually build a couple of therapeutic candidates and that these are actually exciting targets for Parkinson's and a number of other potential oxidized diseases. We're thinking of this as a precision therapeutic platform for multiple diseases. Obviously, focus on Parkinson's first, but we're very interested in, for example, diabetic kidney disease, non-bias, stress and dioxide stress point of view, oncology from a nutrition point of view, and actually, inflammation and a number of age-related other diseases, which I won't go into that. So this is kind of talking a bit more about business. This is how we look at this. So we got a precision therapy approach, mostly for Parkinson's disease. Again, interesting diabetic kidney disease. There's a lot of angles on Alzheimer's disease, which I haven't talked about. Our clinical this platform through redox protein analysis and sits in the right middle of this. It enables essentially cancer therapy development and those and on the other side development of these types of approaches. So with that, I want to thank. I showed you a lot of data. Thank you for your attention, but I really want to just highlight that I'm just a messenger, and I have a lot of help with this. And we've been at this for since 2010. So currently, a lot of the science is done by Balázs Hedberg, who's on the biology side, Balázs Kors, biochemistry and biophysics side. Want to really highlight Max, who actually started, I met him in 2010 here at the chemistry department. He's a PhD student, and we have been really working together since then. So he was a scientist first, and now he's really helping and leading fundraising and a lot of business activities at the company. He's also on the board. Also in the audience is Tom Sawyer, who without him, we would have never gotten started and helped raise seed funding, and has been also looking over development of business and supporting us for long term. He has also a Cambridge PhD. He runs a family office in Hong Kong and Singapore. There's many other people here. I see in the audience Professor Cantabio, but I've also been working for more than 10 years. He's been also very supportive of the Scientific Advisory Board member. And then here is not at Columbia, he's from Purdue University, he's he's SF. These researchers have funded and supported my work and our company's work. And then this is a bit of older picture of the team here. And just to say, there's many other people that have contributed, many different organizations, and this is probably half of the people that the science labs have been working on this. So with that, thank you for your attention, and I'm happy to receive questions and try to answer them.
Thank you so much.
Thank you so much for this very insightful presentation. So now the floor is open for the audience for questions. If you have any questions, please raise your hand. There are microphones and controls. The torch, yes. Thank you very much. Very interesting talk. Thank you. I'm curious about the diseases that you want. Is there any comorbidity of these diseases, or the defect TJ1 or underexpressed TJ1 depend on the requirements of the body? And then how does that then implicate the diagnostic tools that you might be developing? Do you need them samples from different areas of the body, or how is this related? Yeah, there's a number of diseases that region had been implicated, and some of these implications actually are underscored by studies showing changes in regional levels across the whole, across specific cell types. Okay, sure. And I think the critical issue here is that, and it goes back to possibly oxystress and oxidative. There are certain organs, certain cell types that are more sensitive to oxidative stress. And essentially, if you get your redox homeostasis or antioxidant system damaged, then they're more sensitive to oxidative stress increase, and so essentially causes more damage. And there are sometimes they're just a lot more, less sensitive to this. And I think those, and it goes back, those are in those organs or those cell types, you got cell damage can lead to very specific diseases. So when you build your diagnostic tool, would you then need to tailor it for the different cell types you're going to be analyzing? Yeah. So that's the big question, right? So the question is, like in Parkinson's, something's going wrong in the brain. Will you see that in the blood? And I think that has been the billion-dollar question also in Alzheimer's disease, right? And what happened in Alzheimer's disease is that, in fact, some of the full systemic pharmacological tau, for example, leaked out. And can be detected in plasma. Now, the big breakthrough there, everybody knew that you should be looking at disposable tau or some pathological modifications of blood. What happened there is that some ultrasensitive detection methodologies, were developed that enabled really the detection, because these are your very low levels. Now, the EJ1 is a bit different because we got the EJ1 is at low levels, but not as low levels as some of the A-beta or phospho-tau. So we have a sensitive ELISA system, but we don't need ultrasensitive technologies to detect it, which is good because it can be very cost effective. Now, in Parkinson's, we don't know, right? So clearly, there's a lot of regional coming from not just the brain, but from everything else. So when we do our clinical works in Parkinson's, we don't know what's going to happen. But there's a lot of implications that Parkinson's is not just a brain disease, because you have also peripheral symptoms very early on. Okay, so I think I'm hopeful that we'll see something, but we don't know. And this is the main challenge. That's why there's no blood test for Parkinson's, because we haven't, people haven't been able to identify the marker that actually is specific in the blood. That's the hard part. I'll give you another example. Male infertility. So there, in the sperm, you have high oxidative stress and issues. So there is a perfect, because we can do a sperm test, we can analyze the sperm, and we can actually see what happens inside it. And we can have it. That's kind of a liquid biopsy. And so that's easy compared to the brain. So that's why we're also looking at from this point of view, looking at fertility and vitamin infertility. It's a huge problem. It's a huge also market. So yeah, I'm not long answer. Thank you so much.
Hi, that was an interesting talk. Most of the data that you showed with cell-penetrating DJ1 seemed to show that DJ1 is a good prophylactic molecule. You always had it either before nucleation or before you stress the cells. The real question is whether it would be a therapeutic. So when the cells have already got the inclusion, are already dying, will it rescue the cell? And I didn't see any data in that regard. So is this just a prophylactic molecule, or is this going to be a therapeutic tool?
I think that, right, the way you put it, I think it's still therapeutic or preventive, right? Which is very important.
You don't take it before the onset of disease, right? That's the difference.
Yeah, okay. Well, I think just to kind of step back, right? So our aim is not to rescue dying neurons. Okay, that's not our aim, because that's too late. If someone's dying, it's going to be very difficult to stop that. We want to prevent it. So if you look at it, as I mentioned, you have symptoms in Parkinson's after you lost 60% of your substantial matter. So what that means that you're still quite functionable with half of your substantial dopamine neurons around. So if you know this is happening, and you can slow down disease progression, then you're fine, right? So, and that's the easier one. What you're trying to do, what you're saying is, I think it's much harder. So we want to do the easy things first, which is not so easy. About rescuing, so as I mentioned, actually, DJ1, there are some studies suggest that DJ1 is involved in cell survival pathways. So we could actually maybe turn off apoptosis signaling or send a signal for cell survival. So that's also possible. We haven't looked into that, and yeah, we haven't done those models. And you're right, we haven't shown. So we're really looking at it just a preventive. We've been looking at preventive because that's the easier, it's very easier models, much easier models than what you're talking about. But I think I want to highlight that remember, there's no prophylactic or preventive. So this is also a big value at this point, I think.
Have you thought about the practicalities of delivery and how much of the protein would you have to deliver? And are you going to deliver it directly into the brain? Will it then stay localized to the brain, or do you want it to cross the blood-brain barrier?
Yeah, so there's a lot of challenges in delivering proteins to the brain, and for neurodegenerative diseases, there are a number of companies that are taking different approaches. I probably can't answer a lot of your questions, and also we have a lot of data that I cannot tell you now because, I'll give you a couple of examples. Looking at different administration routes, one interesting administration route is nasal delivery because that straight goes into the brain, so there are some technologies there that we're looking at. I haven't shown any in vivo PK data, but if you do an IP injection, there'll be a very nice distribution. So it does get distributed. Brain access is an issue. The brain access, as you can imagine. So I think there's a lot of questions that we're trying to do, and there's a lot of tricks we're also looking at, which I cannot disclose. About levels and all this stuff. These are actually you have very good questions, and these are actually challenges that we're trying to mitigate and get around.
And if you get it into the brain, can you use the blood-brain barrier as an advantage so it can cross into the rest of the body and reduce on-target toxicity?
Yeah, I mean, that's also another question, a good question, because as I mentioned, you increase regional levels, maybe cause cancer. So the idea here is not to overexpress the marker, which is limit missing levels. Okay, and so there's a lot of questions.
I'll just complete this question. Will it make us leave longer healthier if we also address all the ones, diabetes and disease, but also allowing old with all diseases?
Yeah, look, I mean, so on the longevity question, we obviously haven't looked at, and there's not that many studies that looked at lesions role in longevity. As I implied, essentially, oxidative stress, you're going back 50 years, there's been oxidative stress, the theory of oxidative stress and aging, that oxidative stress is responsible for aging. We know that now it's more complex. If you look at a lot of biological processes that are highlighted as kind of driving aging, oxidative stress is unique. And oxidative stress, in some ways, is a biochemical phenomenon. And a lot of times, people look at biological phenomena. So I'll give you an example: mitochondrial dysfunction. It's one of the drivers of aging, but mitochondrial dysfunction. What is mitochondrial dysfunction? Well, actually, mitochondrial dysfunction causes oxidative stress. Oxidative stress causes mitochondrial dysfunction. So it's very much underneath. So it could. So of course, during that, I will say, yeah, sure, it could. But as a scientist, I can say we don't have the proof that it does. We're looking at this from as a therapeutic, and not from a longevity. Look at it from a longevity point of view. It's pretty hard to get funding for that.
All right. I apologize that I'm afraid it's all the time we got for the Q&A. So please join me in yet another round of applause for our speakers now.
That was a very interesting presentation. Okay, thank you for coming. Yeah, actually, I have another question for you. Sure. Just a really quick one. Do you think DJ1 itself can behave like a pre-active?
We haven't looked at this, but we're very interested in looking at DJ1 So one of the things is that because we work in a company setting, DJ1 aggregation is a very academic thing. And so we're actually looking to have a PhD student to kind of look at some very basic physics.
So you want to do some cloud EM to start with?
Well, we want to look at, yes, so we want to look at primary EM, we want to look at liquid seeds, we want to look at, a lot of the basic things. We want to look at what happens in the brain, we need to develop some antibodies specifically to aggregate these ones. So a lot of stuff to do to really properly characterize this.
Definitely. That's very interesting. Because DJ1 itself is so well studied, unlike synuclein or tau or anything like that.
There's a bunch of other proteins that actually, I think of transthyretin. Amylosis is an enzyme, very well structured tetramer, and actually forms a bunch of tetrahydrate amyloidosis diseases.
Absolutely.
And there's actually a form called chaperones that binds to it. It's a very similar concept. No, it doesn't. We don't need to induce her. It's a slow aggregation. So you put tau on the bench, leave it there. It takes 3 weeks for tau to start aggregating. So DJ1 aggregates in about a week and starts aggregating. A couple of days, it's very stable and everything, but it takes a lot longer to aggregate. It doesn't aggregate.
This is